In this mini lecture, we're going to take a look at hearing and equilibrium, which is otherwise known as balance. Before we learn about the physiology of hearing, let's have a little anatomy review. The ear is divided into an outer ear, a middle ear, and an inner ear. The outer ear is the auricle or the pinna, which is a structure of cartilage and skin that is designed to be shaped like a funnel, because this is exactly what it does. It collects sound waves in the air and funnels them into the auditory canal so that these air waves, these sound waves, can be funneled in to strike the tympanic membrane. The middle ear begins at the tympanic membrane. It is connected to the three smallest bones in the human body. These are referred to as our ossicles, and they are known as the malleus, the incus, and the stapes. The malleus connects to the tympanic membrane, the incus connects to the malleus, and the stapes makes a connection to the incus. The stapes also lays up against the inner ear, and the job of the middle ear is to transmit sound waves into that inner ear. The inner ear is made up of a cochlea for hearing, and a saccule, utricle, and three semicircular canals, which will be responsible for balance. Physiologically, the function of the external ear is to collect sound waves and transmit those sound waves to the middle ear. The external ear is known as the auricle or pinna. It is elastic cartilage covered with skin, and it is shaped like a funnel. It surrounds a hole in our skull called the external auditory meatus, which leads to a canal known as the external auditory canal. The external auditory canal is a curved tube of about an inch. It is made up of cartilage and bone. It is carved into the temporal bone, and it ends as the tympanic membrane, or what a lot of people call the eardrum. In order to collect dirt, the external auditory canal will have a thin covering of earwax. Earwax is produced by ceruminous glands, and the actual physiological term for earwax is cerumen. And as I just said, the external auditory canal ends at the tympanic membrane. The tympanic membrane is pretty much the border between the external ear and the middle ear. It is a combination of epidermis, collagen fibers, elastic fibers, and simple cuboidal epithelium. And as its name implies, it's going to vibrate like the skin of a drum. So I've mentioned that the job of the external ear is to collect sound waves. Well, what exactly is a sound wave? A sound wave is a pressure wave of alternating high and low pressure regions. Sound waves travel through air. However, they can also travel through other medium like a fluid. And I want you to think of a sound wave as being kind of like a slinky, being pulled and then pushed and then pulled and then pushed, etc., etc., etc. Because that's in essence what the sound waves are doing in air. Frequency of sound is defined as the number of waves that pass a set point per time period. The relationship is this: the higher the frequency, the higher the pitch of the sound you will hear. In addition to frequency, sound also has an amplitude. The amplitude of the sound wave is the energy or the height of the sound wave. Physiologically, that translates in us as volume, and the relationship is the greater the amplitude, the louder the sound. The middle ear is an air-filled cavity carved into the temporal bone. Its physiological function is to convert sound waves traveling through the air into vibrations of bone. The middle ear is separated from the external ear by the tympanic membrane, and it is separated from the inner ear by an oval-shaped window called the oval window. The tympanic membrane connects to the oval window by three tiny bones referred to as ossicles. There is the malleus, which attaches to the eardrum or tympanic membrane. The incus, which is the bone in the middle. The incus connects the malleus to the stapes. The stapes is the last bone in this order. It attaches by a oval-shaped footplate to a oval tympanic membrane in the vestibule of the inner ear. And that oval-shaped tympanic membrane is called the oval window. The last thing I want to say is that there is an auditory tube which will lead to the nasopharynx. The auditory tube's job is to help equalize pressure on both sides of the eardrum so that we have better clarity of sound. When there is a pressure differential between the external environment and the air in the middle ear, this can dampen how the tympanic membrane and the ossicles vibrate. And this leads to our sound being muffled. Now, one way we can clear this up is we can swallow, because there is a small muscular sphincter that closes off this auditory tube from the middle ear. And when we swallow, that sphincter opens and closes very quickly. And that's going to allow for the equalization of air pressure in the middle ear with that of the external environment. In other words, the air pressure in our oral and nasal cavity. And suddenly when you do that, you will find that the dampened sound you were hearing clears up and what you were hearing is much more sharp and crisp. In anatomy, you probably learned that the inner ear is made up of the cochlea, the vestibule, which contains a saccule and a utricle, and 3 semicircular canals. But really what the inner ear is a bony labyrinth which surrounds a membranous labyrinth. The bony labyrinth is a set of tube-like cavities that are carved into the temporal bone. They are filled with perilymph, and their job is to surround and protect the membranous labyrinth inside. The membranous labyrinth is a set of membranous tubes which will contain sensory receptors for hearing and balance. Some of these tubes are going to be filled with perilymph. Other tubes are going to be filled with endolymph. And truly, the membranous labyrinth is the utricle, the saccule, the 3 semicircular canals with their ampulla, and the cochlea. That's what the membranous labyrinth is. You probably remember from anatomy that the cochlea is coiled up and looks a lot like the shell of a snail. The cochlea is actually 3 fluid-filled canals that roll up to form that snail shape. These 3 fluid-filled canals are known as the scala vestibuli, the cochlear duct, and the scala tympani. The idea is that the stapes, which is vibrating up against the oval window, is going to send vibrations into the fluid of the scala vestibuli, scala tympani, and cochlear duct. And it is these vibrations in fluid that ultimately are going to trigger the neurons for hearing. The fluid pressure that builds up in the cochlea when the oval window bulges in will be dissipated by the bulging out of another window called the round window. And when the round window moves in, the oval window presses out. So the fluid pressure in the cochlea does equalize so that it does not damage the neurons of hearing. If you were to section a piece of the cochlea, you would see this picture here. You would see that there are 3 fluid-filled canals. On top, there's the scala vestibuli. On the bottom is the larger scala tympani. And sandwiched in between these two is a fluid-filled canal in the middle called the scala media, also known as the cochlear duct. The scala vestibuli is separated from the cochlear duct below it by a membrane that I'll draw in red here called the vestibular membrane. The cochlear duct is separated from the scala tympani below it by a membrane known as the basilar membrane. And sandwiched between these 2 membranes will be a very unique accumulation of neurons, which is the organ for hearing, referred to as the organ of Corti. Within the cochlear duct is the organ of hearing. It's called the organ of Corti, named after Alfonso Giacomo Gaspari Corti in 1851, but it's also known anatomically as the spiral organ. Now, the separate cells responsible for hearing are called hair cells. These hair cells have stereocilia that can bend. Their bases are anchored to the bottom of the spiral organ, which is right here. It's called the basilar membrane. And the stereocilia are embedded inside a big gelatinous membrane here called the tectorial membrane. The bending of the stereocilia is going to result in graded receptor potentials. Now, before we go, I want to point out that there are two kinds of hair cells. We have the outer hair cells, which are long and thin. So there they are, there's their stereocilia. And then you also have inner hair cells, which they look a little bit more like a bulb. I'll draw that there. But again, they have stereocilia. So we're going to see in the next couple slides the way this works. Because the stereocilia are the triggers, if they bend one direction or they bend the other direction, you may actually get depolarization and it results in a graded receptor potential. Why this happens? It's going to have to do with fluid pressure, and it's going to have to do with the movement of the basilar membrane with respect to the tectorial membrane. And that's what we're going to see in the coming slides. Located within the organ of Corti will be two kinds of hair cells. There are the outer hair cells, which are longer and thinner. I'll circle one right here. And then there are the inner hair cells, which are a little bit more bulb-like in their structure. The outer and inner hair cells synapse with first-order neurons. The first-order neurons are shown in this picture in yellow. Now, depending on what textbook you read, these first-order neurons will be considered to be pseudo-unipolar neurons. Some textbooks call them bipolar neurons. It doesn't matter what kind of neuron they are, the first-order neurons synapse with the hair cells via these bulb-like structures called afferent terminals. And I'm going to circle 3 of them right here in green. These are known as afferent terminals. They form synapses with the hair cells. The cell bodies of these first-order neurons are going to cluster together to form what is known as the spiral ganglion. And then the axons leading out from the spiral ganglion, these axons are collected up to form the cochlear branch of cranial nerve 8, which you can also call the cochlear nerve. I have a couple pictures in this slide, again, to show you the organ of Corti. The picture in the upper right, you can see here circled in blue, are going to be the afferent terminals of the first order neuron, which is communicating with these hair cells shown in yellow. And these afferent terminals are sending out long axons which collect up to form the cochlear nerve. And down in the lower left-hand corner, this is a model that we have in our anatomy classroom. It shows, again, the organ of Corti with the vestibular membrane on top. Then you have the tectorial membrane and the basilar membrane sandwiching the hair cells, forming the organ of Corti. Okay, let's learn some of the physiology that underlies hearing. We're going to start off with the auricle, the external ear, collecting sound waves, funneling them into the auditory canal. That's step 1. Step 2, the sound waves are going to hit the tympanic membrane, causing them to vibrate with the same frequency and amplitude as those sound waves. Step 3, the ossicles that are attached to the tympanic membrane are going to vibrate with the exact same amplitude and frequency of those sound waves. Because the malleus is attached to the eardrum, the malleus transmits this vibration to the incus, and the incus transmits those vibrations to the stapes. And because the stapes lays on top of an oval window, now in step 4, the vibration of the stapes up against the oval window is going to cause the oval window to vibrate. And the vibrations of the oval window are going to become vibrations of fluid within the inner ear. In step 5 in this diagram, movement of the oval window is going to create waves in the perilymph fluid we find in the scala vestibuli. This fluid wave is going to be propagated along the scala vestibuli. It's going to go all along the length, and then it's going to turn the corner at the apex, and it's going to continue into the scala tympani. And that fluid wave is going to propagate along the scala tympani. As the fluid moves through the scala tympani, it's now going to cause the round window to bulge out to help dissipate that pressure. So that's actually step 9 in the diagram. But let's go back now to step 6, which is the fluid wave propagating along the scala vestibuli and eventually into the scala tympani. As the fluid moves along the scala vestibuli, it's not only pushing forward, but it's pressing down into the cochlear duct. My analogy for you is you taking a rolling pin and rolling it along a length of dough. You're not only rolling forward, but you're pushing down on that dough. So the rolling pin is your fluid pressure. The dough would be the cochlear duct. And as that fluid is moving along the scala vestibuli, pushing forward, it's also pushing down, and eventually that pressure wave pressing into the cochlear duct is going to cause the movement of the basilar membrane within the organ of Corti. That's going to depolarize hair cells, and we're going to get a graded potential. Question is, how does pressure wave in the scala vestibuli become pressure in the cochlear duct? Remember, my analogy is you are rolling a rolling pin along dough, and the rolling pin is the fluid pressure in the scala vestibuli, so that's moving forward, but you're also pressing down on the dough, which is the cochlear duct. And the idea is that pressure, which is now going to be transmitted into the endolymph fluid in the cochlear duct, is going to cause the basilar membrane to vibrate. So this basilar membrane, as the fluid pressure moves, is going to move back and forth, back and forth. Because the hair cells are connected to the basilar membrane at their base, the base of the hair cells are going to move back and forth with this fluid pressure. And that is going to bend our stereocilia back and forth, back and forth. The bending of the stereocilia happens because they are embedded in the tectorial membrane, and the tectorial membrane does not move. It is the basilar membrane which shifts back and forth. The tectorial membrane remains steady. That means the hair cells are locked in there, and as the base of the hair cell moves, the stereocilia bend as well. When you bend a stereocilia a certain direction, they are going to open up mechanically gated ion channels, and you are going to get the influx of positive ions into those hair cells. Believe it or not, most of the ions moving in are going to be potassium. I know you're used to depolarization of a neuron being caused by the influx of sodium ions. This is an exception. So our hair cells are going to depolarize because potassium ions from the endolymph are moving into those hair cells. We're going to depolarize up, but we're never going to reach our threshold. We're going to stay below threshold and create a graded potential or a receptor potential. That's okay because the graded receptor potential opens up voltage-gated calcium channels in the hair cells, and that influx of calcium causes neurotransmitter release from the hair cells. So our hair cells never reach threshold. They don't reach an action potential, but that's okay. They can still release their neurotransmitters. Those neurotransmitters, that is glutamate. Glutamate is going to bind the first order neuron, shown here in this picture in yellow, and it's going to trigger an action potential in those neurons. If we were in the classroom, I would have drawn this picture on the board. So since we're an asynchronous class, we're going back to our virtual whiteboard. And in this virtual whiteboard, we have a drawing of both our inner hair cell and our outer hair cell, and how these two hair cells communicate with the afferent sensory neurons. One thing I haven't added to this diagram that you may have noticed in the previous slide was the fact that there are motor neurons that also communicate with these hair cells. Those motor neurons were shown in blue in the previous slide. The afferent neurons were shown in red. I'm going to be honest with you, I don't know really what these motor neurons do, and I only have a limited amount of information in my brain. So that limited amount of information is going to be devoted to the afferent neurons. Because I don't really know what the motor neurons do, you don't have to either, and you can just ignore them. All right, let's take a look at the figure. We have an outer hair cell, we have an inner hair cell. We're going to bend the stereocilia in one direction. This is going to result in the depolarization of these hair cells. They are going to generate a graded receptor potential, and that's going to result in the release of neurotransmitters. Neurotransmitters are then going to bind to the sensory neurons that are forming synapses with these hair cells. Specifically, what they're going to bind are these little black knobs that I've drawn here. These are called afferent terminals. They are going to extend out as peripheral axons. Peripheral axons are often called afferent fibers in textbooks. So the action potential is going to be generated in these afferent fibers. Neurotransmitter is going to bind the terminals. It's going to depolarize these afferent fibers to threshold. You're going to get an action potential. And that action potential is going to head down the peripheral axons. The cell bodies of these pseudo-unipolar neurons, because that's what they are, the cell bodies are going to cluster together to form a spiral ganglion, and then you are going to have axons coming out of those. These are going to be your central axons, and they are going to travel into the central nervous system as the cranial nerve 8. Specifically, again, it's going to form the cochlear nerve of cranial nerve 8. These axons are going to enter the central nervous system at the medulla oblongata, and then you are going to continue on with the remaining part of this sensory pathway. Our first order neurons in the auditory pathway make a synaptic connection with the hair cells, and they do so through these little bulb-like afferent terminals that I've shown in previous slides. The axons of these first order neurons are going to form the cochlear branch of cranial nerve 8, which you may have learned as the cochlear nerve. Now, these axons are coming into the brainstem. We're going to form the first part of the auditory pathway, but the auditory pathway isn't your typical 3-neuron pathway. It's actually 2 pathways made up of multiple neurons. I want to concentrate on the one shown in red because it's a little less complicated than the one shown in green. So we've got our cochlear axon terminals stopping in the medulla oblongata. You have a choice. Your first order neuron can stop in this nucleus right here called the cochlear nucleus. Or you can have a first order neuron stopping in that nucleus and then connecting to the other side of the medulla via a smaller extra first order neuron. And that's going to stop off here at the superior olivary nucleus. So in essence, you kind of have 2 first order neurons. You have the one coming from the cochlea entering into the medulla, and then you could have a secondary first order neuron, a little mini first order neuron, which crosses over to the other side of the medulla. Those first order neurons are now going to connect up with second order neurons, and there can be multiple second order neurons. Again, let's concentrate on the one on the right-hand side shown in red. We have a second order neuron connecting here at the superior olivary nucleus, running up into the midbrain and stopping in the midbrain. Then there's a little mini second order neuron that runs from the midbrain and stops in the thalamus. So even though there are 2 second order neurons in this section of the pathway, they do what second order neurons do, and they end up in the thalamus. The last part of this pathway is a third order neuron, which is going to do what a third order neuron typically does. It's going to run from thalamus to the cerebral cortex. This one specifically ends up at the primary auditory cortex in the temporal lobe. So while we have a couple extra first order and second order neurons, they kind of do what they're supposed to. The first order neurons come into the brainstem. The second order neurons eventually end up in the thalamus, and the third order neurons eventually end up at the cerebral cortex. If you can understand the concept of hearing, the bending of stereocilia causing the opening of mechanically gated ion channels, the flow of positive ions in resulting in the release of neurotransmitters, then you understand also the idea of equilibrium, because the physiology of equilibrium is the same as the physiology of hearing. It involves neurons called hair cells. It involves fluid pressure changes, bending their stereocilia, so there's a lot in common. This is an anatomy review slide that reminds you about the structures of balance. Physiologically, we have 2 kinds of balance called static equilibrium and dynamic equilibrium. Static equilibrium allows us to maintain the position of the body by basically knowing where the head is relative to the force of gravity. The hair cells responsible for static equilibrium will be found within the structures known as the saccule and utricle. The saccule and utricle on this picture are right here. Here's our saccule. And our utricle should be right next to it there. They are located in a region of the inner ear called the vestibule. Dynamic equilibrium allows us to maintain our body position by knowing where the head is when it's moving. So it's basically the detection of any kind of movement of the head, rotational movement, deceleration of the head, or acceleration of the head. The hair cells of this, they are called cristoreceptors. They're going to be located within the ampulla of a semicircular canal. So here, right here on this picture is an ampulla of a semicircular canal right there. And you guys know from anatomy, we have 3 semicircular canals. Let's start off with static equilibrium and learn the physiology of the saccule and the utricle. Within the saccule and utricle, there are thickened regions called a macula. There are 2 macula per inner ear, one in the saccule, one in the utricle, and they're perpendicular to one another. When you look at a macula, you'll see that it contains 2 types of hair cells. You can see it's got a skinny hair cell, it's got a fat little hair cell. You can see the hair cell has their stereocilia coming off of them. You can see that the stereocilia are actually embedded in sort of this membrane. In this case, the membrane is called an otolithic membrane. You can see at the other end of the hair cells that there are going to be afferent terminals. They are basically going to be the ones, I think in this picture, they are the ones in blue. And they, again, are going to come up and form synapses with these hair cells so that when the hair cells release their neurotransmitter, these afferent terminals can bind onto those neurotransmitters and then stimulate their action potential and perpetuate that into the central nervous system. So there's a lot of similarities between a cochlea and a macula. I want to tell you a little bit about the otolithic membrane because it's a little different than a tectorial membrane. It is a little bit looser and a little bit more gelatinous. It's secreted by the supportive cells of the macula, and it contains these calcium carbonate crystals called otoliths. And these otolith crystals will move when you tip your head one direction versus another. When you move your head, it's going to lead to the movement of these otoliths. So the otoliths are going to kind of shift back and forth, back and forth. And when they shift back and forth, they cause the bending of hair cells, the stereocilia of hair cells in a particular direction. This is going to open up mechanically gated ion channels. You're going to get in, again, a sub-threshold receptor potential. So you're not going to depolarize to threshold. And that depolarization is going to be caused primarily through the movement of potassium. So if this sounds really familiar, it's because it's pretty much the same mechanism that we used for hearing. The interesting thing is that if you bend the stereocilia in one direction, you're going to increase ion flow into the hair cell, and you're going to increase the receptor potential rate, meaning you are going to get increased release of neurotransmitters when these stereocilia bend one way. When you move your head back to the original position and the otoliths shift back to their original position and the stereocilia swish back to their upright position, this rate of depolarization slows, so you don't get as many receptor potentials and you don't get as much neurotransmitter release. And that low level of neurotransmitter release, which is going to cause action potentials in these neurons in blue to set up, well, that's going to be ignored by the brain. It's only when you move these stereocilia and these neurons right here increase their rate of action potential firing that the brain actually wakes up and says, oh, what's going on? Oh, I must have moved my head. So tilt your head forward or tilt your head to the side. You get a bunch of action potentials firing out of your saccule and utricle, and your brain goes, huh, that's normal. Oh, that's not normal. Oh, my head's moved. And when you put your head back up to the original position, the saccule and utricle decrease the number of action potentials they fire, and your brain basically goes back to ignoring what the saccule and utricle are telling it. The saccule and utricle have 2 types of hair cells that are called type 1 and type 2 hair cells. The type 2 hair cells are going to synapse with their first order neurons via bulb-like structures called afferent terminals. There they are right there. These are the afferent terminals that I've described also in the cochlea. The type 1 hair cell, the cilia is going to bend. You're going to depolarize. You are going to generate a graded receptor potential. You are going to release neurotransmitters. These neurotransmitters are going to bind onto the afferent terminals and stimulate an action potential in this first order neuron. The afferent terminals are also seen in connecting up with our type 1 hair cells. But in addition to this connection, type 1 hair cells connect to the first order neuron through a second kind of afferent terminal, which forms a little cup around the bottom of the type 1 hair cell. I'll diagram it here in blue. This little cup-like structure is called a calyx. It's going to do the same thing an afferent terminal is going to do. It's going to bind neurotransmitters. So the type 1 hair cell, when you bend its stereocilia, you're going to depolarize it, generate a graded potential. The neurotransmitters are going to get released, and these neurotransmitters are going to bind onto either the afferent terminal or the calyx. That's going to stimulate an action potential in those afferent fibers. All of these action potentials that are being stimulated in this first order neuron are going to travel along these fibers. They're going to pass through the vestibular ganglion, which is the cell bodies of these bipolar neurons, all clustered together. And then that action potential is going to continue out the axons leading out the other end of the vestibular ganglion. And those axons cluster together to form the vestibular nerve, or the vestibular branch of cranial nerve 8. These action potentials, just like in hearing, are going to head into the medulla oblongata, where the rest of the sensory pathway for equilibrium will continue. Dynamic equilibrium, or the balance of movement, is going to be detected by the semicircular canals. Specifically the hair cells, which are going to function the same way they do in static equilibrium and the same way they do in hearing. They're going to be located within the ampulla at the base of each of the semicircular canals. Remember, we have 3 semicircular canals: anterior, posterior, and horizontal. They're going to detect the movement of the head in different planes of movement. Contained within the ampulla is going to be a structure called a crista. A crista is a bunch of hair cells covered with a gelatinous sort of dome-like structure called a cupula. I guess in architecture, a dome-like structure on a house is known as a cupola. Notice that our hair cells right here have their tectorial membranes embedded in this cupula. Fluid is going to flow through these semicircular canals in either direction. So I bet you any money you can understand what's going to happen now. When you move, the fluid flowing through the semicircular canal is going to go through the ampulla. It's going to flow over the crista. It's going to bend that cupula gelatinous dome, and it's going to cause the bending of the stereocilia, and it's going to act just like static equilibrium and just like hearing, in that the bending of these hairs in one direction is going to increase the release of neurotransmitters from these hair cells, thus increasing stimulation of our first order neurons. There's something there called an ampullary nerve. I think all of those ampullary nerves gather up again to form the vestibular branch of cranial nerve 8. The first order neurons form the vestibular nerve, and they end in multiple vestibular nuclei. The second order neurons will come out of these vestibular nuclei. They will connect up, they will run up to and end up in the thalamus like your standard second order neurons do. Specifically, they stop in the ventral posterior nucleus of the thalamus. And then the third order neurons go from the thalamus to the cerebral cortex, specifically to the temporal lobe, where you have the vestibular area of the cerebral cortex. So that's this pathway right here. It is your standard 3 neuron, first order, second order, third order pathway. But you can see from this picture that there are a lot of other neurons coming out of the vestibular nuclei in the medulla oblongata. So there are other pathways that can emerge, and many of them are motor pathways. For example, the vestibulospinal tract, the one we talked about in the efferent motor pathway. This is the tract that adjusts postural skeletal muscle contraction in response to head movement by using the cerebellum, input from the cerebellum and input from the motor cortex. Now, if you remember correctly, the spinocerebellar tract started off with sensory information coming into the medulla. We had those upper motor neurons, and then we had the upper motor neurons and the lower motor neurons. Well, that sensory information is this equilibrium pathway that you see here on the slide. There are other second order neurons that will run from the nuclei. Instead of going to the thalamus, though, they will end up at the other nuclei in the brainstem, specifically the nuclei that are the origins of cranial nerves 3, 4, 6. These are the motor nerves that will control eye movement, and also the nuclei of cranial nerve 11, which is the accessory nerve. These are involved in neck movements. So in essence, you have a little bit more of a complex system, but at the base of this equilibrium pathway is your standard 3-nerve sensory pathway. By the way, you notice that there are two terms, vestibular ganglion and vestibular nuclei. The vestibular ganglion is in the peripheral nervous system, and it is associated with the inner ear. Don't confuse that with the vestibular nuclei, which are found in the medulla oblongata. So that's just one last thing I wanted to tell you, because some of you may be going, wait a minute, you just used the word vestibular nuclei and you talked about it in the inner ear. I used the term vestibular ganglion. All right, so we've done hearing, we've done balance. We are now going to move on to the last lecture in our special senses. This is actually going to be the last lecture in nervous physiology. It's going to be mini lecture 10E, and it's going to deal with vision. So do yourself a favor, take a little bit of a break, give yourself a little bit of rest and relaxation time, and then come on back and we will do vision. For those of you that are wearing contact lenses, I'm going to just give you a heads up. If you want to help preserve your vision, you may want to take your contact lenses out when you're looking at fine print like that on a computer screen. Why? Well, I'll tell you in the lecture that we have on vision.